The goal of this video is to highlight the key features of the Watson and Crick structure of DNA. On the left side of the screen, you can see the nitrogenous bases as they are categorized by their molecular structures. The weak hydrogen bonds between the nucleotides, seen on the right side of the screen, are what keep the two strands of DNA together. This diagram is used to describe the concept of base stacking. Here, the nitrogenous bases interact via hydrophobic bonding of their pi cloud electrons. These forces, as well as the van der Waals forces, help keep the long strands of DNA intact. The phosphate backbone of DNA refers to the phosphate group attached to the deoxyribose sugars via phosphodiester bonds at the 5' carbon of each nucleotide. These phosphate groups are also responsible for the negative charge of DNA. Sugars of nucleic acids, often referred to as pentose sugars, have 5 carbons. In RNA, a hydroxyl group is attached to the 2' carbon, whereas in DNA there is only one hydrogen at this position. The extra oxygen atom in RNA makes it more reactive and less stable than DNA, making DNA the better option for storage of information in the long term. The attachment of bases to the backbone sugars through glycosidic bonds is asymmetrical. This results in the formation of two different grooves on opposite sides of the base pairs, the major and minor grooves. The major grooves occur where the backbones are far apart, and the minor groove occurs where they are close together. This diagram is meant to help understand the handedness of the DNA coiled helix. The normal form of DNA is exclusively a right-handed helix, so it is important to be able to distinguish between right and left-handedness. It helps to imagine this image in the upright position, but if you do, that front-facing strand is clearly exhibiting a left-handed helix. What I do that helps me differentiate between the left-handed and right-handed coils is decide which strand is on the upper side of the coil, and if it's the strand moving to the left on the upper side, then it means that it is a left-handed coil, and if it's the strand that moving to the right-handed side is on the upper side, then it means it's a right-handed coil.